Hello, listeners of this lecture. This lecture is intended for students of the Biology Educational Program. I am Kabataeva, Senior Lecturer of the Biology Department of the Sәрсен Amanjolov, East Kazakhstan University. Theme of our lecture: the rules of Mendel and their application. Purpose of the lecture: to form the concepts of Mendel's laws, monohybrid and the dihybrid, crossing, gene, genotype, and phenotype. We will consider the next questions. First, the principle of dominance, monohybrid crossing, the principle of segregation, the principle of independent assortment, dihybrid crossing. The main laws of heredity were first discovered by the Czech scientist Gregor Mendel. Gregor Mendel was a young priest, a science and math teacher. Mendel, in his publication called Study with Plant Hybrids in 1886, stated that hereditary material isn't a combination of body cells but the combination of unchanging units called genes. And these units are inherited from generation to generation independently and make new combinations. Also, the importance and validity of these statements are still true. They were not accepted and understood in his life. Later, De Vries, Correns, and Charmak proved the statements of Mendel in their works. These scientists published their findings in the title Laws of Mendel in 1900. Gregor Mendel, who is the father of genetics, used garden peas to demonstrate important principles of inheritance. While planting and harvesting pea plants on his monastery, he noticed patterns of traits in pea plants. Most pea plants turned out to have green pods. Some had yellow pods. Some had yellow seeds, and while others had green seeds. Stem length, petal color, pod shape, location of flowers, all these seemed to exhibit a pattern of inheritance. Also, pea plants have a lot of features which are available for crossing. These plants produce 5 generations in a short time, have many different types, have genes that are independent. The genes found on different chromosomes control different traits, have reproductive structures completely enclosed by the petals. Self-fertilization is easily prevented, and the plants are easily protected from other sources of pollen. Mendel went on to breed pea plants to see how these traits acted. Through these experiments, Mendel created 3 laws that govern how traits are passed on from parents to offspring. At the beginning of the experiments, Mendel shows true breeding, individuals carrying homozygous genes, true breeding lines exhibiting opposite features of the traits. Mendel's studies proceeded as follows. He prevented self-fertilization by removing the male anthers from the parent plants. Then he transferred the stamens of flowers exhibiting the opposite feature to the stigma of the flower, whose stamens had been removed earlier, and ensured the plants to cross-fertilize. Mendel observed that the offspring produced from this experiment resembled only one of the parents. Mendel repeated the experiment many times, which gave the same result. That is, the offspring, F1, resembled each other and one of the parents. Mendel demonstrated this by crossing peas with yellow and green seeds. The cross between two individuals in which only one trait is being studied, called as a monohybrid cross. When performing genetic analysis, a certain rule is used to record different hybridization schemes. Parent forms are denoted by the letter P. Sign of the female sex is symbol of the planet Venus. Sign of the male sex is symbol of the planet Mars. Sign of hybridization, X. Gametes symbol is letter G. Generation symbol is letter F. And the first generation is designed at F1, the second generation F2, the third generation F3, etc. For any given trait, an individual inherits one gene from each parent so that the individual has a pairing of two genes. We now understand the alternate forms of these units as alleles. An allele is one pair of genes, one coming from the mother, the other from the father, in the chromosomes of a living thing. Alleles can be similar or different. Alleles of a particular trait are at the same location on homologous chromosomes. These gene pairs control one trait. When peas carrying these two traits were crossed, all F1 individuals were the same as each other and yellow. In these types of crosses, the features that appear in F1 are called dominant. The features that aren't seen are called recessive. In crossbreeding, dominant features are expressed with capital letters. Recessive features are expressed with the lowercase form of the same letter. If the two alleles that form the pair for a trait are identical, then the individual is said to be homozygous. And if the two genes are different, then the individual is heterozygous for the trait. Determining and recording the gamete type are very important in genetic studies. Genetic crosses cannot be made without knowing the gamete type. Therefore, before crossing the subject, the genes that form the traits and determine the types of gametes should be known. Pay attention to the example. Phenotype ratio, all seeds are yellow. Genotype ratio, 100% heterozygous. Genotype, the complete set of genes of a living thing or the genetic makeup of organisms. Phenotype is the physical, visible characteristics of organism. Green seed, for example, yes, brown eye. The phenotype of a living thing is determined by its genotype and environment. From this experiment, Mendel concludes that when two alternate forms of the same gene are present in an individual, normally only one, the dominant allele, is expressed, became visible on phenotype. This is principle of dominance. For the next stage of the study, only first-generation peas were used. Mendel planted it and left it without intervention so that the bees could pollinate themselves. This allowed the second generation of F2 plants to appear. Due to self-pollination, yellow and green seeds appeared, and trait yellow is the dominant feature. Pay attention to the example. Phenotype ratio 3 to 1, genotype ratio 1 to 2 to 1. This experience helped to establish that the trait of one of the parents, green, didn't disappear completely, but was simply inactive or suppressed. From here comes the second law of Mendel under the name the principle of segregation. When gametes are formed in meiosis, the two alleles of each gain segregate from one another, and each gamete receives only one allele. The hybrid cross is a cross between two organisms in which two different traits are being studied. Let's see a cross between homozygous dominant yellow round-seeded pea plants and homozygous recessive green ringlet-seeded pea plants. In the first generation, the plant acquired the following characteristics. So, yellow color and round of seeds. The phenotype ratio at F2 is 9 to 3 to 3 to 1. This ratio was 3 to 1 in monohybrid cross. So 9 yellow round, 3 green round, 3 yellow wrinkled, and 1 green wrinkled. The result shows that the transmission and inheritance of two different traits don't depend on each other. Alleles for one characteristic behave independently from alleles for other characteristics during gamete formation. When two or more traits are examined in a single cross, each trait is inherited without relation to the other traits. This occurs because the alleles for each trait are sorted into the gametes by chance, independently of one another. All possible combinations of genes will thus occur in the gametes. This is the principle of independent assortment. The Mendel's findings can be summarized as follows. Traits are inherited through genes. The traits of a pea plant are related to its alleles, which can be the same or different. If the pair of alleles forming a gene are different, the effect of the dominant allele appears in the phenotype. The recessive allele remains hidden. Variety in organisms arises from the fact that during fertilization, the gametes combine randomly, allowing the genes for various traits to form new combinations. Traits are visible in offspring in predictable ratios according to the principles of probability. So, in this point, our lecture is over, but I won't give you for self-work the next